Good afternoon, everybody. My name is Elise Myers, and I'll be talking to you today about complex lipids and microbial mats and stromatolites of Hamelin Pool, Shark Bay, Australia. So I know it's been a long day, so I'm going to break down exactly what we're going to talk about first. I'm going to go into defining the field site, defining what microbial mats and stromatolites actually are, then why would we even study diverse lipids in Shark Bay, and then why study the ones I focused on in particular. Then I'll go into the experimental procedures in terms of the technology and the analysis that I used, as well as the process itself. Then I'll go into the compound-specific results, conclusions, and future work. Okay, so to start off, Hamelin Pool, Shark Bay, Australia is located in the western part of Australia. It's this part here. So it's a very isolated U-shaped loop. And because of this isolation, it's very hypersaline. So it's already shallow, about 9 meters in depth overall. There's not much river and input. It's very arid climate out there. And there's also not much contact with the outer ocean. So this hypersalinity is a really important factor of Shark Bay. It's also a really important factor in why there are stromatolites and microbial mats. So if you think about it, stromatolites and microbial mats, well, microbial mats themselves are this kind of combination of organic material, kind of filamentous bacteria that are coming together, that are trapping sediments and forming this kind of like a layer that you could actually peel back in some places when it's centimeters thick. And because of this hypersalinity, it actually prevents a lot of other organisms that will come in and eat this delicious feast of organic material. And then it actually allows them to grow. And in Shark Bay, we can actually see this picture here, I think, is very indicative that in Shark Bay, there are these microbial mats that are on top trapping these sediments. And then the bottom part is actually carbonate precipitation to make these stromatolites, which are the rock-like formations that we can see. And the importance of studying these is that they are a potential modern link to ancient forms of life. Stromatolites have been found all over the world, especially some of the oldest ones in Western Australia and in South Africa at about 3 billion years old. And if we're able to understand what the microbes are in these forms right now, so we can understand a little bit more about the community diversity in the currently forming microbial mats and stromatolites, then we can understand a little bit more about the environmental conditions and the composition in the ancient past. And that leads me to why we would actually study these diverse lipids in the first place. So if you think back to your beginning biology and chemistry classes, the external membrane for cells is there in order to protect them from everything that's outside of them, so from salinity, pH, temperature, those kind of things. So if those external environmental conditions change, the membrane lipids are going to then adapt in order to protect the cell just as much. So by seeing this, the adaptation of membrane lipids over time, then we'll be able to understand a little bit more about the environmental conditions both then and then also comparing them to those that are happening right now, which we can understand and see very clearly. And then they're also able to show microbiological diversity. So they're like characteristic fingerprints. So if you think about it, you're taking a microbial mat and you're taking out all of the lipids that are within it. And then with those lipids, we can now correlate that to whatever microbes are there. So it's a really good way for us to understand what exactly is living there. And it's able to also help us to see in terms of what proportions. Because remember, microbial mats have a lot of sediment in them, so we're able to see how much is actually biogenic material and how much is correlated to each type of microbe. It also is going to be, this study is an improvement of the current characterization of lipids, because lipids, as we know them right now, we have a general sense of what their function could be, but there, of course, as is wont to happen in science, there are theories and then they are disproved in the next 10 to 15 years. So this will be actually very helpful for understanding a little bit more about what these lipids signify and what the function is for these lipids themselves. And the bottom line, why we would study multiple of them is studying multiple lipids would then give us a better understanding, a more complete picture of what is actually happening. So the different lipids that I studied were fatty acid methyl esters, and then intact polar lipids, glycerol, glycerol, dialkyl, glycerol tetraethers, GGTs, which is what we'll use from now on, and then bacteriophoane polyols. So I ordered them, the second three here, in terms of increasing persistence or recalcitrance. Mind you, we are not in 19th century literature class. Recalcitrance here means persistence in the rock record as opposed to obstinate children. So why would we study each one of these lipids? There's a very important reason for studying each one. So intact polar lipids. These are lipids that actually have the polar head groups still attached to them. And polar head groups have a lot of different extra characteristic fragments on them. So like saccharides and trimethylamines, those are actually the separate head groups. And those can be really easily identified with the microbes that are there. Well, they're more characteristic than a lot of the other ones. So they're especially helpful for characterizing living microbes and what's there right now. And then the glycerol, dialkyl, glycerol tetraethers, GDDTs, I actually looked at them in two different forms. So in the GDDTs, which just means that it has the glyco group attached to the top. So that's the intact polar lipid form. And then the GDDTs, or the GDDTs, those ones are the core lipids. And those ones have lost the polar head group, but have higher persistence. So looking at both of those together, it actually offers a really cool glimpse at both living and long deceased microbes. And it's very easily comparable because the two of them are one and the same, essentially, or from one and the same. And then fatty acid methyl esters. These ones were added because they're highly studied and they have well-defined mass spectra. So by looking at these, we'll be able to understand what is actually going on there. And also, these were studied before by Michelle Allen in the same exact location on just the pustular and smooth mats. And she had very well-defined mass spectra for the fatty acid methyl esters. So in doing this again, I got to see whether or not I was doing it right. And it was a very good check on my work. And then the bacteriohopane polyols are the last ones that I looked at. Those are highly recalcitrant, again, persistent. And those are really well preserved in the rock record. And those are extremely important for if we're trying to make that link from these modern microbial mats to the ancient stromatolites, because that's what we'll be able to see in the ancient forms. So how did we identify each one of these lipids? And I want to just bring it out in the beginning so that we all understand where we're coming from. So GCMS, gas chromatography-mass spectrometry, that was used for the fatty acid methyl esters in order to identify those compounds. And I'll go into exactly what you get out of that in a little bit. And then high-performance liquid chromatography, or HPLC system, we used it coupled to a QTOF machine, so it's a quadrupole time of flight. And basically, from all of that you need to take is that offers MS. So we can put in the TLE, so the total lipid extract, and then ask for the instrument to pull out a specific mass, which it then does. And then then it goes through and looks at what are the dominant signatures at that particular mass. And then it fragments those. So we can see at the different fractions of that lipid there. And those characteristic fractions are actually how we can determine if that's the lipid that we're looking at. And that's this last part down here. So the published mass spec fragmentation patterns. And how you can think of that is to think of like a long lipid chain. There's going to be a place where that's going to be more willing to break. And you're going to see a higher signature where it's going to be breaking more often. And if you see that, then that's what you can relate then to that is that lipid because you saw it broke at that spot most often. And then for other checks, of course, the exact mass was used. That was one of the first parameters. And then retention times in terms of relation to commercially available standards were used just to double check. And then the final check was this MS fragmentation. So now I can finally go into what I actually did. So I unfortunately did not get to go to Australia, but I got the freeze-dried samples, just as good. And so I had 12 samples. So I had 5 smooth microbialites, 2 on the top, 2 on the bottom, 6 colloform microbialites, and then 1 pustular. So what I mean by 2 on the top, 2 on the bottom, the samples are about 5 centimeters in depth. And then the ones that were on the top were then from 0 to 5 centimeters. The ones on the bottom are 5 to 10 centimeters. So it gives a really good look at what is that microbial community and how is it changing over depth. And here's just the diagram on the side. That'll be really helpful because I actually found a lot of connections to morphology. So it'll be interesting to look at that later on. So if you see the smooth mats there, you have this very laminar parallel structure, very filamentous bacteria-based. Whereas the colloforms, you're adding in a little bit more coarser grain sediment, so you're going to have a little bit more laminoidal, a little bit more wavy formation. And then for pustular, you have this irregular clotted formation. OK. So in the experimental procedure that I went through, it's got quite a few steps. I won't go through all of them, I promise. So we start out with this sample up here. So that was the freeze-dried microbial mat sample that I was given. And that was then ground up with mortar and pestle using geoclean products. Everything was geoclean, which means that it was subjected to cleaning through a couple of different solvents to remove all organic materials so that we got no extra external signatures. And then it was subjected to Bly and Dyer extraction. So it's basically a way to make sure that we get all the total lipid out of the microbial mat itself. Then the total lipid extract itself was divided into 3 separate areas. One spot was sent over to look at intact polar lipids and the GGTs, so both the intact form and the core lipid form. The other portion was then taken to bacteriohopane polyols. And then a third portion was subjected to fraction work. So this is the TLE here, so you can understand a little bit more what exactly we were looking at. So from that microbial mat before, after extracting all of it, we could blow it down and get this very thin amount, small amount of TLE. It's about 0.15 to 0.25% by weight of the whole microbial mat itself. And then columns on the side there, that's how this process went through. So you use a different combination of solvents in order to elute out different compounds. So you start out with your most nonpolar compounds that come out and then going up to polar. And the ones that we're looking at in particular were the fraction 3, the fatty acid methyl esters. So how were results quantified? Because it's really good to understand what is there, but it's actually very important to understand in what abundance and how much. So this is achieved through a combination of internal and external standards. So internal standards being things that were put inside, different compounds that were put inside the actual sample itself to see how well did I extract the total lipid extract. With the injections, how much were we actually getting. External standards that were used to calibrate the instruments, as well as overall portions from sample and total lipid extract. So from the amount that was initially taken, each time there was an injection that was done, it was only a small portion of that amount. So then I had to keep a running table of what is actually, how much of the TLE is this, then we can multiply and figure out, okay, of the whole mat, this BHP is then this percentage or this amount. And here's an example of a quantification method that was achieved through the MS-MS. So this is just another way to visualize what's happening there. So this is one of the extracted ion chromatograms. And so basically, this is where we've told the instrument to pull out at a certain mass. And this is for the immunotrial BHP. And so then from that, I ask it to extract another extracted ion chromatogram for the most specific mass, and then that tells us exactly what that compound is. So it's for that exact amino trial. Then you integrate under the area, integrate under that peak to find the area, so that's the relative amount that you would have. But in order to quantify that, you also have a standard that is subjected to the same exact analysis. So you have a standard that then has this same peak. I extracted from that, found the overall area. So then the relationship you have is that 100 nanograms, or however much amount of whatever standard it was, in relation to its peak area, was then equated to a certain number of nanograms of particular compound, in this case the amino trial BHP, divided by the peak area that was found for that one. So that's just how those were quantified. So now we'll go into the actual results, now that we understand what everything is, where it's coming from. So for the first lipid that I analyzed, that was the intact polar lipids. And these ones, as I mentioned before, have this very characteristic polar head group. They are really great for determining the composition of and quantity of viable biomass. So remember before, there's a lot of sediments inside these microbial mats, but it's good for us to understand how much is actually lipid and how much is actually alive. And something to note about these is they quickly degrade after cell death. They have been shown to degrade at about the rate of a couple hours to a couple days. So these are really good for looking at currently living or recently deceased microbes, as opposed to ones that have been dead for a long time. So this is a heat map. This is the results that I got from this experiment. And this is just a fairly typical distribution. So I put this up here just to show you what we would get. And I noted a few on the key there. Those are ones that I think you should look at in particular. And I'll talk about what their significance is. But here, to understand it a bit, this is your elution time. So think about it like the columns that I showed you before. This is essentially just how the machine runs them out. And then the mass to charge ratio there is essentially, for all of my experiences, was just the mass. Because the charge that was on all of the ions that I was looking at was H+, and it's just in an ion form because that's what the instrument can recognize. So what is the significance of those results, of all of those many lines everywhere? They are very relative results. It's not an exact quantification. So this is just the initial step to understand what is in the sample. So there's a high abundance of DDTS and DDTA, which you can just call your betaine lipids. And then that shows a prominence of algae and non-vascular plants and fungi. And then your ornithine lipids were also very prevalent. So we'll look again. So that's your DDTS and your DDTA. And then your ornithine lipids, your trimethyl ornithine lipid, as well as your ornithine lipids. And those ones actually signify slightly different things. So your TMOL, trimethyl ornithine lipid, that's for your anaerobic autotrophic metabolism, those types of organisms. And then ornithine lipids, they have this possible connection to sulfate-reducing bacteria. I say possible because only in the last couple of years has that been really talked about a lot more, and there have been a couple papers that have agreed on it, but the last one was in 2013, so who knows what could happen. And then from the IPLs, the biggest things that we can take away is this non-vascular plant or lower plant signature, as well as these anaerobic autotrophic microbes and the sulfate-reducing bacteria. So we have to figure out, what about amounts of that? So then that took us to the glycerol, dialkyl, glycerol tetraethers, the GGTs. So these are the membrane-spanning lipids. A lot of times when we think about lipids and membranes, we think about the lipid bilayer. But in this case, think about your cell, this is the outer membrane, and these ones just span the membrane itself. And the core lipids persist, so the ones that have lost that extra glyco group that's the polar head, those ones persist for a bit less than 140 million years. So they can last for a long time, and they give us a whole new time window that we can look at. They're highly indicative of microbial origin, so when we see them, it's very exciting. We know that that's what's happening. And they have been used in the past as dynamic environmental parameter proxies. So essentially, this here is GDT 0. There are 0 cyclic rings within it. So if I had GDT1 up here, you'd see a 6-member carbon ring within that structure there. And when you add in that other ring, it actually increases the stability of that molecule. And so you'll find ones with more rings in them in microbes that are more like heat tolerant and things like that. So that's why they've been able to be used for those kind of proxies. Because if you see the disappearance of GDT0 or GDT1, you know that the conditions have become too extreme to have just those signatures anymore. And how they were identified in this experiment was through different ions, the ones in bold, so H plus and NH4 plus, those are the ones that I looked at. And that's just asking the instrument to select out for those masses when you have those compounds. So this here is an H plus ion one. So you see in the corner over there, OH2 plus, so you have that ion, that's what was found for this one. So what did we actually get from these? This is a relative abundance for the GGTs. And this is the second round because the first round was done in order to figure out what concentration we needed to have them at in order to get a really clear signal. So if you run through and remember the peaks that I showed you before, that's kind of the information that you'd get. But in the first round, there was a lot of noise, and the signal was not extremely high in comparison. So then that data was not good quality data. So I concentrated it 5 times in order to make sure I had a really high signal-to-noise ratio. 3 to 1 is the absolute minimum. And we attained that this time. So these results are much higher quality and can be trusted. So something to note right away, GGT 0. So that is this darker blue that's here. There's a stronger dominance within the smooth mats than the colloform mats, which suggests dominance by anoxygenic phototrophic organisms. And that, I thought, could then be related to the morphology. If you remember back to those pictures that I showed you before, the smooth microbial mats don't have a lot of, they're just kind of like little layers right on top of each other, the filaments. And I thought that maybe that could be related to this dense filamentous bacteria of smooth mats not allowing as much oxygen diffusion throughout the mat and thus promoting microbes with anoxygenic metabolisms. And then this is actually supplemented by the other finding, which is the Krenarchaeal GDT. This is the light blue there. And the Krenarchaeal GDT relates to ammonium oxidizing thalmoarchaea, so things that need to have oxygen for their metabolism. So then there's a higher amount of those within the colloform mats as opposed to the smooth mats, which again could be lead right back to the morphology, but in the opposite sense in that there's more oxygen diffusion, so then there's more of these microbes that can be there. For the colloform mats overall, something that you should note is that they had the highest amount of, oh, sorry, that's the next slide. Oh, yes, the elephant in the room. That's what I wanted to address next. So I know that you guys probably all see this giant red bar in the top one that is so uncharacteristic of the other ones. Unfortunately, I cannot tell you very much about that because GDT1, which is what that one is mostly studied in terms of its disappearance when the temperature becomes too high. So remember what I talked about before with the environmental parameter proxies? That's what that's mostly studied for right now. So there's not much that we can really say about what that could be because it's very unlikely that the temperature is much different between the 2 layers of the mat that are a couple centimeters apart. Okay, so then this is a more quantified look at this. So the other one was relative abundance. This is now where I've gone through and I've quantified the actual amount and then I've normalized it of the nanograms of GGT of each one in particular to the grams of the total lipid extract, of the total amount of lipid that I took out from everything. So from this, big things that you can take away, well, actually first, caveat that this is all combination of the samples. So there were 5 samples of colloform that were able to be quantified. There were 5 samples of smooth mats. So these 2 have the same amount of samples. This was only the 1 sample. So when I'm averaging them, I'm dividing everything, these ones by 5, and then this one is just maintained the same. So for the core GGTs, the ones that lost their polar head groups, those are so much more abundant than intact GGTs. So if you just look at the x-axis here, you see that this one for GGTs, core GGTs, that is closer to 5,000, whereas for the glyco, glycerol, dialkyl, glycerol, tetraethers, those ones are in between 2,000 and 2,500. So there is more dominance within the microbial mats of these deceased organisms as opposed to the ones that are currently living. Something else to note is that the colloform mats have the highest average amount of GGTs, and so when I divide everything through, they still have the highest amount for all of them, and that then suggests that there's higher microbial density in the colloform mats themselves. And then the smooth mats have a lower portion of GGTs, so the currently living ones, which I thought then could lead back to morphology potentially, but it's essentially showing the unfavorable environmental conditions, so there's just less of the microbes that are currently living right now in the smooth mats in comparison to all the other ones. And something to note here is that the colloform mats and pustular mats actually have fairly similar distributions. So then that thought of morphology being one of the big factors is confirmed with that, or not confirmed with that, is supported by that, because pustular mats also have that ability for oxygen diffusion a little bit deeper. So the next lipid that was analyzed, now that we understand a little bit more and we know that there is evidence for these ammonium oxidizing ones, were the fatty acid methyl esters. And as I mentioned before, these ones were analyzed in 2010 by Michelle Allen. So this was mostly a check on my work, and then also let's see what else we can find out from these profiles based on the additional information that we collected from other lipids. So fatty acids in samples are taken to fatty acid methyl esters, which have fairly stable degradation products. So they're subjected to treatment by methanolic HCl, so HCl plus methanol, heated for about 3 hours in order to change their form to the methyl ester form. There are well-established profiles, as I mentioned before, and they are identified by this characteristic fragmentation that I mentioned before. And here's a clear diagram so you can see exactly what I was talking about, where things break at a certain spot more willingly. So you'll break here, and that is your characteristic McLafferty rearrangement ion. So that is what you see the most often with your fatty acid methyl esters. That's what really confirms that that's there. They also break at 55, 87, 101. So there's just a couple different fragments that you look for in order to confirm that that's what it is. So here's a fairly standard plot of the results that I got for the fatty acid methyl esters. So just to understand the key a little bit more, NC14, NC anything else is your saturated fatty acid methyl esters. So they have no double bonds within them. And then your double bonds are denoted here. So 16,1 has one unsaturation within it, and then 18,2, which was seen there, that has two unsaturations. And then also down here, there's a combination of your anti-iso compounds, iso compounds, as well as branched fatty acids. And because this is your fairly, this is the distribution that you get most often for the samples that I looked at, I want to talk about a little bit what this means. So there's a dominance of C16 and C18 peaks, with a few exceptions in a couple of the different plots that I had. But this is fairly characteristic. This is in agreement with Michelle Allen's paper. It's also in agreement with other samples that have been collected from Hamelin Pool, like UIDs by Roger Summons in 2013. So this is actually a good check that we see that there's a lot of dominance by those. There's also a 10-methyl C16, which is highly indicative of sulfate-reducing bacteria. And we're not sure if this NC16 is actually that. And to do that, we'd have to elucidate a little bit more about the chemical structure itself to figure out if that's actually what we're seeing. There's also this significant C61 and also this significant C18 one. And it'd be really interesting to understand exactly where those unsaturations are, because that could tell us a little bit more about what that lipid is and what it's doing there. Additionally, there are these larger carbon chains. So if you see down at the bottom, there's C22, C24, C26. There's this even over odd preference, and that is related to detrital organic matter. So then that's indicating that there's a possible terrestrial source that's bringing in these leaf waxes that could be ending up there. However, keep in mind that Shark Bay, remember we talked about it being a really arid environment. There's not much river input, so it's probably not coming by rivers. It could be coming as an aeolian source, so being brought in by air. And then the short chain, odd carbon numbered branched fatty acids. Those were the ones that are located down here, a couple of those. And those ones are likely bacteriophospholipid, and those are really highly indicative of sulfate-reducing bacteria. So then from our fatty acid methyl esters, we have even more support that there are these sulfate-reducing bacteria there. And we're on our last lipid. So the last one that I analyzed was the bacteriophospholipid polyols, and these are the ones that are highly recalcitrant, so they can be seen very far into the rock record. They have an undefined function, but they're likely for structure. So they have really specific side chains, and then having these specific complex side chains leads to rigidity within them. So then there's, if they're incorporated into the membrane, the thought is that they would then increase the rigidity of the overall membrane itself. There has been tied to a lot of different samples, a lot of different functions, but then of course it has been debunked multiple times. So it's hard to understand exactly what they're for. There is recent evidence though. Paula Wellender in 2009 actually found that they might be really helpful for reducing membrane permeability to protons, which then would make them helpful for pH resistance. So overall for the membrane, they're there. We're not exactly sure why, but that's possibly one of the reasons. And they're also relatively unknown in marine cyanobacteria. The hopanoid production there is relatively unknown. So BHP is also called hopanoids, so you know. And most of the hopanoid studies have been actually in freshwater ecosystems. There have been a couple in marine systems, and especially in Shark Bay in particular. But so then this offers a really interesting look then, and a really interesting study in diversity of BHPs within marine systems. So what did we get? This is on a log scale, so you know. So there's a clear dominance of BHTs. About 74% to 94% of the BHPs was BHT. This is a good thing. If you look at a lot of different other studies, the dominance is about 80%. So we were right in the boundary there. So these samples were likely run correctly, and the information can be trusted. And the next thing that's really abundant is amino trials. So those are these orange bars here. And those ones are indicative of purple non-sulfur bacteria. However, they also have been linked in the past before to terrestrial inputs. So those are kind of conflicting viewpoints, and again, this kind of goes back to where there's a little bit of wiggle space in the field of understanding exactly what they're for. But something that I noticed was that the amino trials were a bit more dominant in my samples that were on the top. So potentially that means that this is from the terrestrial, or potentially it's supporting the terrestrial input as opposed to this purple non-sulfur bacteria, sorry. Then there are also these 2-methyl BHTs. These are ones that were found by Michelle Allen in 2010 also. She was the first person to actually find them there, which was very exciting. So then we've seen them again and confirmed their presence there. And these ones are actually related to cyanobacteria. They were thought initially to be just cyanobacteria. That was just proven that they were produced by other organisms. But a study by Garby in 2013 identified the different microbes that live in this area. And the 2-methyl BHTs were attributed to cyanobacteria. So we can say that this is a strong signature, a strong indicator of cyanobacteria. And then last one, 3-methyl BHTs. I promise you it is worth both of the punctuation marks that are there, because these have not been found before in these samples, which is extremely exciting. So the 3-methyl BHTs, we'll go back to this diagram so you can understand. They are going to come out at the same mass. The 2-methyl BHT, 3-methyl BHT is only a difference in the placement of the methyl group itself. So when scanning everything, it comes out at the same mass, but it's going to elute at a different time because you're going to change the polarity by having the methyl group in a different place. So you have your 3-methyl here and your 2-methyl there. So in confirming these, we can't say 100% that that's what they are, but because they eluted out a minute later, and because they have the same characteristic fragmentation of the 205, which was, I'm sorry, let me point that out in the graph here. So if one of these is a methyl group, then the characteristic fragmentation is 205, and that's what I saw repeatedly. Also, it was seen in 9 out of 12 samples. So all of this compounding together suggests that this is really what they are. But in order to confirm that, we'd have to get an organism that produces 3-methyl BHTs and then compare what it produces to what we found to make sure that that's actually what it is. And then this is just to help you visualize the abundance of the BHPs overall. So as I mentioned, the BHT is extremely dominant, and you can see that here. Also, do note that the 3-methyl BHT, it's actually a noticeable portion of each of the samples where it's found. So I'm really excited to hope that that is what it is, and hopefully we can confirm that at some point. So conclusions. The lipids showed fairly characteristic patterns, things that we expected to see from based on Michelle Allen's paper and also based on other studies of the microbes in the area. So far, so good. And then there's also evidence that we've seen for ammonium oxidizing and sulfate-reducing bacteria. That was from multiple of the different lipids that were studied. So that is highly likely that those are the microbes that are living there. And we got to understand a little bit more of where they came from, because if you look at the data, and what is talked about in my thesis a little bit more is the difference between the different layers, and then we can understand where there's a little bit more concentration of sulfur-reducing ones, where there's a little bit more concentration of ammonium oxidizing bacteria. There's possible presence of 3-methyl BHTs, which I can't emphasize enough how exciting that is, and hopefully that's what they are. And then they have been linked recently that the genes for their production have been found in aerobic environments, so potentially that's what this is signifying, but there's still not a whole lot of information on where 3-methyl BHTs come from. So this could actually be something to bolster the field a little bit, in that if they were found here, we can understand a little bit more that this is the type of environment that they were found in. And then also created a thorough lipid profile for the marine microbialites, stromatolites, and microbial mats that were here, which was something that wasn't done in all of these lipids before. And then there's potential influence of morphology, which I found particularly exciting. So I think that that's something that warrants further study for sure, but it's, I think there are some clear signatures and clear patterns that are occurring between the differences in the morphology of like pustular and colloform mats versus smooth mats. And then there's potentially this minor terrestrial input, which would be interesting to understand where it comes from exactly. Which leads into future work. And everything in bold here is stuff that I'm doing currently right now. So the fatty acid methyl esters, if you remember, we had those giant signatures of 16-1 and 18-1, so they have the one unsaturation, and we have no idea where that is. And that's why we're treating it with dimethyl disulfide in order to find out where exactly that unsaturation is, and then understand a little bit more about why those lipids are there and what they signify. There's also going to be further analysis on the hydrocarbon and the polar fractions. So if you remember, the fatty acid methyl esters were fraction 3 of a 5-fraction separation there. So we're going to look at fraction 1 as well as fraction 4 and 5. And then hopefully, this one should be like italicized. Hopefully we'll get the culture before I leave so we can compare and see if we can confirm the 3-methyl BHPs. And then I think that it'd be really interesting to have further sampling of the pustular mats, because the pustular mat that I had was just one sample as opposed to the separated into different depths. And I think it'd be really interesting to understand the oxygen diffusion and how that works in terms of different depths, especially in relation to colloform mats, to see like what is the difference between the two of them there. And then, of course, which the overall goal is comparing these signatures from these modern lipids to ancient stromatolites and understanding a bit more about the environment in which these stromatolites were forming and a bit more about the community dynamics at that time. So I'd like to give very special thanks to Roger Summons, my thesis advisor, who took on an undergrad who wanted to bite off a lot more than she could chew, to Flo Schubatz, who is my postdoc and who was absolutely fantastic when I was in the lab and also has been reading everything I've written from Germany. And then, of course, to Emily Mades, Julio Sepulveda, Keldon Pear, and Carolyn Colinero, who guided me through the lab, understanding all the instruments, understanding the compounds. The E25 GGG groups who adopted me as an undergrad, and to the whole family there. And then also, of course, to my friends and family, a lot of my friends who are here right now, and I thank you so much for coming to support me. It really means the world to me. And so thank you for all of your attention, and I will do my best to answer any of your questions.